So
Sir, your slide is visible,
my slide
but
is
it is
visible
not.
in slide mode, no?
Yeah. Your slide is visible, but it's not
Yes.
changing.
Slide is moving, sir. Slide is moving.
No, sir. No, sir.
Slide is moving, sir.
No, sir.
No, sir, it's not moving.
Yeah. Okay. So basically, so chromatography, if we see, the chromatography is an analytical method that compounds are physically separated prior to measurement. If we have to measure or quantify the compound, we need to separate from a complex mixture. The main purpose of chromatography is to separate from a complex mixture or detect from a complex mixture and quantify the target sample in the matrix. So the chromatography could be like a normal liquid chromatography, gas chromatography, or supercritical fluid chromatography. So today our discussion will be mainly focused on the liquid chromatography. So if we see we have to use, we can use the HPLC. HPLC is a very important analytical tool in the different sectors of science. It has a very high resolution and it has a very high sensitivity. Like we can detect the compound in a ppm and ppb. So if suppose any of compound is present in C, it has a good repeatability. We can reproduce the experiment, whatever the experiment is being done, and a small sample size is required. Even for like if I have a glass chromatograph, like previously we are using, we need a huge amount of sample. But many times we see the sample size is a limiting factor. We get very small size, like in a natural plant product, we get very small sample size. So that sample size could be a determining factor or limiting factor. But in HPLC, small size sample we can use in analysis. It has a very high sensitivity. And moderate analysis can use in analysis because in gas chromatography high sensitivity. We need to vaporize moderate analysis can use in analysis. But many gas chromatography vaporization vaporize analysis can use in analysis. Many gas chromatography vaporization vaporize gas
Your slide is not changed. 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 So it is not moved. Your slide is not changed. No, sir. No, sir. So it is not moved. Your slide is not changed. No, sir. No, sir. Yeah, it is not moved. Now it is there. Your slide is not changed. No, sir. No, sir. Yeah, it is not moved. Now it is there. Your slide is not changed.
If we see the scope
Yeah,
of
it is not
HPLC,
moved. Now it is there.
like in pharmaceutical, if we see the antibiotics and reduced like in pharmaceutical, if we see biochemicals like proteins, carbohydrates, like feed enzymes, food product, especially the toxins, mycotoxins, which affect the food quality, or food additive, or the preservative. So coloring agent. So if these coloring agents are safe or not safe for the food safety and safety, it is very important. We can detect that harmful chemical or useful chemical. Industrial chemicals, forensic chemistry, like many time in solving the forensic cases, like what kind of drugs or poisons or the blood alcohol or narcotics, the HPLC can be used. Environmental field, now it is getting great due to the environmental pollutant which is being continuously being poured in our environment and affecting the human health and the biodiversity and environment. So this, in environmental field, the scope of HPLC is very enormous, and the clinical medicine, like bile acid drug, drug metabolites, how it is being metabolite and what quantity urine extract. So the scope is very wide. So if we see a simple flow diagram of HPLC, a mobile phase which is being pumped by normal injector or auto sampler. Nowadays auto sampler is being used in a column, and there are detectors of various kinds. So just I tell that why HPLC, like it is a simple column chromatography. It is it is an analytical method in which the solvent is being forced in a very high pressure, 6000 psi, very small particle size of the column packing material, and much greater surface area for interaction. So like very small sample, it is pushed into very high pressure column, and very small size particle of the column make the interaction between analyte and the adsorbent, mobile phase and the solid phase, stationary phase and the mobile phase interactions. These allow much better separation of component of mixture compared to normal chromatography or column chromatography. The major improvement over the column chromatography concern the detection of method which can be used. These methods are highly automated and extremely sensitive nowadays. So if we see simple like a chromatogram, that like if the sample is being injected, that a simple peak we get, and this peak, what is its height, what is its retention time and area. These are the important parameters which are being considered in HPLC. So some of the important terms like what is chromatogram, just I saw. What is mobile phase? Liquid phase that move the solute through the column. Stationary phase is the packaging material in the column, which is immobile phase in the chromatographic phase. And the peak, the visual representation of chromatogram based on the detector's electrical response due to presence of sample component in the flow cell. So whatever the chemical we want, whatever the component we wanted to detect, this the peak is very important. And retention time taken by analyte to reach the detector, how much time. So it depends on the size, it depends on the polarity, so how much time it takes to do. So this HPLC is both qualitative technique, means we can identify the component from mixture, and both quantitative technique, means we can measure what amount of the component, what is the amount of the component in a part unit. So what is the basic separation mechanism is that the components are separated because the molecules in a liquid phase or in a mobile phase, they move at a different rate in the column. Like suppose if we have a two component like red and the blue, which is shown in the column, they will move at a different rate in the column, depending on their size, depending on their interaction with this stationary phase. So this first one will be separated first, maybe on their due to their size, less interaction with the column, they will move fast. Later on, the second component will be separated. So this is a very simple principle on which how much time it is taking to interact. So due to different interaction between stationary phase and different sample, molecule moves at a different rate. So those who have a stronger interaction with stationary phase, they will get eluted later, and those has a weaker interaction, they will be eluted first. So this is a basic separation mechanism. So based on the separation modes, even column liquid column chromatography can be classified into a normal phase chromatography, reverse phase chromatography, which is being normally used in the lab. In normal chromatography, the column is generally plugged with the silica, which is a polar in nature. But in reverse phase chromatography, the column is plugged with the hydrophobic molecule like ODS, octadecyl silica. So this in ion chromatography, it is based on the charge, and size exclusion chromatography based on the size of molecule, and affinity chromatography based on the affinity of the molecule. So different modes are there, and we can see in a normal chromatography, packing material is here in the column is silica, and in reverse it is a silica or octadecyl silica, and the mobile phase in general being used as a hexane or IPA in the normal phase chromatography. But in reverse chromatography, like methanol water or acetonitrile water, so here the attraction is adsorption, and here interaction is a hydrophobic. In size exclusion is a porous column, mainly the mobile phase is a tetrahydrofuran and gel permeation. Ion exchange chromatography, ion exchange gel buffer solution and ion exchange affinity chromatography packed with the ligand based on the buffer solution, and it is based on the affinity. So these are some of the types of the chromatography. So but the reverse phase is the most widely used. C18 column, C8 column, these are the columns which are most widely used in the day-to-day lab detection. Other, depending on the need, depending on the necessity, they are being used. So the stationary phase is a non-polar, ODS, and the mobile phase is a polar, like methanol plus water, acetonitrile plus water. This combination is being defined as the reverse phase chromatography. So you can see these are in the stationary phase in reverse chromatography, octadecyl silica type, octyl type, C4 butyl type, phenyl type, TMS type, or cyanotype. So these are the variant form of the column. You can see on the silica C18H37. It is like big hydrophobic molecule, like a fatty acid, like big hydrophobic molecule. So that's why in reverse phase column there is a hydrophobic interaction. So stationary phase is a non-polar property, while the mobile phase is having a polar property. So mobile phase, what are the like a solvent? Water plus buffer, an organic solvent, mainly methanol, acetonitrile, THF, and the buffer is being used phosphate buffer, acetate buffer, and other. Ratio of aqueous to organic solvent is very important. In later phase of my lecture, we will see how this changing ratio, just changing the ratio of organic solvent and water, could how they can separate the very near peaks from the thing. So if we see that what kind of interaction, like see the less polar analyte, more polar analyte. So the less polar analyte will interact more with this column. So they will be eluted later, and those more polar analyte, they will be eluted first. So less polar analyte, more hydrophobic analyte are more attractive and spend more time in the column, while the hydrophobic bonded phase, therefore they will be eluted last, while those have a polar in nature analyte, they will be coming first in the peak. So hydrophobicity, if we see, if the sample has a more like this kind of like carbon chain or aromatic chain, they will spend more time in the column. But if the sample has like a carboxyl group, amino group, hydroxyl group, which has a weaker hydrophobicity, they will spend less time and they will be eluted first. So this is the, we see this, if like a phenol is there, so it will have a stronger interaction with the ODS C18 column. So it will be eluted later, but those have a polar or less hydrophobic, they will be eluted first. So we can see more hydrogen bonding or other kind of bonding is there. So if we see, like just I say that how can be the mixture of the solvents if we have like water and methanol into 20 to 80 ratio, 2:8, then you can see the peaks, like we see the hydroxy methyl benzoate, para-hydroxy ethyl benzoate, para-hydroxy propyl benzoate, para-hydroxy butyl benzoate. These compounds are very much similar in their molecular structure, no little change in the like ethyl, methyl, butyl in the chain, but basically they are very similar. So they will be eluted, but they will be eluted very clump or very near to each other. So this will be very problematic. Suppose those who are working on a preparative mode, those who want this compound to get purified or as a single component. So from this kind of peak, it will be very difficult. We cannot elute. But suppose by changing the ratio of water and methanol 30:70 or 40:60, you see they are getting these components which are very similar in nature, just change in a few like ethyl, methyl or propyl group. By changing the ratio, we can elute this component separately and we can use for the analysis. So increasing the solvent polarity is one of the factor that being very important in the HPLC. Like you see the effect of stationary phase, like strong interaction, like C18 have a medium interaction and C4 has a weaker interaction. So depending on our goal, what we wanted, like in HPLC, it is not like a magic box. No, we need to know that what is our sample. We need to know our sample. We need to know our what kind of column we are going to use. So sometimes we think that the HPLC is a magic box. It is not so. It is a scientific tool, and how we can use that tool, that is very important. Sorry. So this solvent type, just I already tell that in reverse phase we use this kind of the solvent like water buffer, acetonitrile and methanol, and the compound type neutral to ion, non-ionized compound which can be dissolved in water and organic mixture. In ion pair reverse phase, same as above, in addition of ion pair reagent, ionic or ionizable compound again. Normal phase, organic solvents, solvent type we use organic solvent, and the mixture of isomers or compound not soluble in organic or water mixture. So in ion exchange, water plus buffer, inorganic ions like protein, nucleic acids, or organic acid, and here in the size exclusion chromatography, water, tetrahydrofuran, chlorofuran. So the high molecular compound, if our choice that we wanted to separate high molecular compound, then the size exclusion chromatography could be your choice. So just in a nutshell, like gel permeation, size exclusion, gel permutation, normal phase, reverse phase, ionic phase, depending on what is their molecular weight, depending on the polarity, we can choose what kind of high performance liquid chromatography we can use. So reverse phase, you see the reverse phase has a white, so non-ionic polar compound, even water soluble compound. So you can see the reverse phase is in a median value. Like in normal phase, you see here, so say, which has a silica column, ion exchange from gel exclusion. So that's why the reverse phase is being most widely used compared to other. But yes, what kind of thing you wanted to achieve in HPLC, that is very, very important. So depending on that, you can choose. So if we see what as an instrumentation, we have seen the principle of the column or principle of mobile and steady phase. Now component, one by one we will discuss about the components of HPLC. Like a modular HPLC has a possible configuration: solvent delivery pump, sample injector. Nowadays column oven is very, very important, which can maintain the temperature has a important impact in the separation mechanism and the detector. So these detectors, column over, sampler injectors, nowadays automated HPLCs are coming, and the delivery pump. So like one pump, previously being only one pump is being used, now binary, now quaternary. So depending on what kind of pressure you want to achieve, like in quaternary we cannot go very high pressure because we have to mix the 4 solvent at a time, 3 solvent at a time. So the binary pump we can achieve great pressure, but we cannot mix many solvent at a time. Two solvent we can use at a time. So this solvent delivery pump and the injector and the column ovens, they are become, so these form the integrated HPLC. So this possible configuration, like isochromatic mode, solvent concentration in isochromatic mode HPLC, whatever the solvent concentration you are choosing, with the time frame you cannot change. It will be remain constant throughout the constant. But in a gradient phase, like in a 5 minutes, 20 raised to 80. In a next 5 minutes, 30 raised to 70. In a next 5 minutes, 70 raised to 30 raised to 70. So you can choose what kind of solvent with reference to time. You can in a machine you can give the command, so it will gradiently, you can increase or you can decrease depending on your solution. So isochromatic mode and the gradient mode, these, like you see in the isochromatic mode, the concentration of the solvent remain constant, but while in the gradient mode you can increase and decrease, and depending on what amount you want to mix, so you can. So this is the elution time. Like in an isochromatic mode, like it is a long time analysis. Like suppose methanol and the water in a 6 raised to 4, you have separation. In isochromatic mode, suppose this like 8 raised to 10, the separation may be very bad separation and it will take 2 time. So but if suppose you have chosen the gradient, you see the concentration is keep on increasing. Methanol concentration keep on increasing with the time. So this give a better resolution compared to isochromatic. So it depends that how, whether you if your isochromatic mode is also giving better separation, it is okay. But if it is not giving, then you have to go for the gradient mode. But with one pump, we can, with simple pump, this isochromatic mode is only choice. But if you have a 2 pump or 4 pump, then you can go for the gradient. So if we see this isochromatic system, a mobile phase is there, a pump is there, an injector and column and detector. Simple system with one pump and one solvent reservoir. It is a isochromatic mode. If more than one solvent is used, solvent need to be, yes, you can run, but solvent need to be pre-mixed itself. One solvent you can use, but if you want to use 2 solvent, 3 solvent, you have to pre-mix. The concentration will not change with the time. But you see the low pressure gradient. Low pressure gradient, one pump used to control 4 reservoir. Mixing is done to a valve. It will check. So one pump will be there, and this pump will be injecting, will be controlled, and it will be injected in the HPLC. An online degasser is very necessary, because while mixing there is a very high possibility that gas may be formed. But nowadays, high pressure gradient like quaternary, binary, pump, many pump companies are coming with many pump. Like solvent A is there, it will be controlled totally by a pump. That particular pump. B will be mixed by another pump. C will be mixed by another pump. So what will happen? The mixing will be more homogeneous with the, because one pump, it will not have it cannot generate so much of pressure on the column. The higher amount of gradient of pressure you cannot generate. But if the like binary and quaternary pumps are there, then you can mix and you can achieve excellent gradient accuracy. And 2 to 3 pumps are required. One pump per solvent is used, and online decaying may be not very much critical in such kind of a thing. So that's why nowadays binary is very popular. Yes, quaternary also, depending on your situation, people are using the quaternary pumps. So if we see the instrumentation principle, how this is mechanically it looks like, the solvents and the pumps, and it is being column, and this is the detector, and this is the illusion. But in a very sophisticated, these are the like a detector and the thermostated column, auto sampler, binary 2 pumps, vacuum degazer, and the solvent reservoir in the bottom. So solvent before we mix like a solvent, there are solvent inlet filters, because whatever the solvent we are getting, many times it is not so pure. No, it is not 100%, even 99% pure. So there may be impurity. So to avoid the impurity, because many times this impurity may impact your column, not only the column, but it will impact your separation. It will impact your detection. So these kind of the solvent inlet filters in a bottle form in our laboratory, we use that one. And apart from that, there are guard columns. The guard columns which will protect your analytical column, but it will reduce the pressure. These guard columns are there, but it will impact the this motor, this pump. So depending on, but at least one guard column is very important, because these analytical columns are very costly, 40 to 50,000, and even if some impurity will go, it will clog, your column is gone. So that's why the guard column is one of the important factor. And this is a simple degasser. Degasser are needed for removing the gas. Suppose you are using binary or quaternary simple one pump to mix the solvent. So degasser may be needed from the solvent, removing the gas for the solvent before they are mixed and compressed by the pump, especially during low pressure mixing done by quaternary pump. A gas can be bubble. As a result, baseline measured by detector can drift, a noisy retention time, and peak area may be not so stable. So this degasser unit. So these are the HPLC pumps. So like even pumps are also there. Their pumps are like constant pressure pump. As the name suggests, they provide constant flow rate in the columns, and they use the low pressure gas contained in the cylinder create desired high pressure. Syringe type of filter, they use motorized syringe to get a constant solvent delivery. Changing the voltage motor, different flow rates can be obtained. And reciprocating pumps, which is nowadays, they consist of hydraulic chamber where the reciprocating piston is placed. This way solvent get in the pump. When the pistons move back and push into the column, the pistons move forward. So it is widely used due to a reason that good pressure generation, constant flow rate, and useful for both gradient and isocratic mode. And space occupancy is also low compared to other pumps. So if we can see here, reciprocating pumps. This is the diagram how motor, particularly plunger, and this inlet and outlet. So this is so these are the low pressure pump. This pump is most critical, high pressure gradient pump and low pressure gradient pump. So the pump is most critical piece of equipment for successfully operating the HPLC. Performance parameter of HPLC pump like flow precision, flow range, delay volume, and the pressure pulse and the composition precision. So this is very important. So we can see here the high pressure mixing binary pumps. The mix point occur at a mixing chamber which is located past the pump, and this is characterized by the lower dwell of volume system. This lower dwell of volume characteristic allow binary liquid chromatography system to change in the gradient much more rapidly than the column, allowing for faster recuperation. So this dwell volume is called as gradient delay volume of gradient online mixing system. Is the volume of liquid contained in the system between point where gradient is formed and the point where mobile phase enter into the column. For the gradient kind of the thing, this particular kind of the thing is very important. So low pressure mixing quaternary pump. So here you can see the proportionating valve. So the point where mixing of mobile phase begin is a right after proportionating valve. So the accounting dwell volume in a low pressure is all this volume mix take into consideration, and this volume from valve to pump head and the pump heads, the volume of pump head and the volume of pump heads to column. So additional injection holding loops is required. Any additional mixing that are added to facilitate further mixing. This leads to larger dwell volume of the low pressure system, which can lead to longer time recuperation condition each column after the sample size. So obviously the various, you can see the various hurdles are there. So obviously the mixing will be less, and this kind of, but it depends what kind of the things we are doing, and like high and low, they have their own limitation, own merits and the limitation. So like when HPLC was there, this kind of injectors like loops front injection, so previously, but nowadays it is being automated sample injector manual, like previously how is been there, but nowadays these auto injectors are being coming here. Without any syringe or loop, you can directly put your sample in this tray or box, and like 96, in our case we have a 96, so 96 sample you can load and you can give the command. So this auto injector are, and see many time in a like a manual injection, precision may be not very much high. It may be appropriate amount will be not there, but in this auto injector the precision is very high. Whatever command you have given, it is automated, so obviously the precision is very high. The simple diagram, how particularly step 1 step 2. So injector, the principle of auto injector. So this rinse, manual priming, rinsing port, measuring, and this it is the sample. So once it is there, so it will rinse, it will prime, again it will rinse, and the measuring pump, and then it will take from the wall, and then from the wall a sample, it will go to the sample loop, and then from sample loop it will go to the high pressure valve, and then it will reach to the column, and then to the detector. So there is a, like in a auto sampler, there is a, well, like whatever the sample while it is being taken, it will be rinse, prime, and rinse, and then again to the, so it is very highly precise. So apart from the auto injector, nowadays the column oven is also one of the very important attachment. And like in the last decades, the column constant temperature for solvent column is required to perform reproducible. Many times you are working, suppose this column is not there, today's temperature 30 degree, next day 40 degree or 45 degree, and because nowadays we are facing very much high heats, so every day your result will vary with the temperature. So for such kind of the thing, suppose if you are on a system you are working which is not much vary with the temperature, but it is not going to be a very obvious thing. It will obviously, temperature will have its own impact, but how much impact, that is very important. If it has a negligible impact, then it's okay. But if you see in a daytime, in a night time, with the change in temperature, there will be different. But if suppose temperature is constant, then you can get your resolution or the peaks in a very smooth manner. So this temperature will have a very impact. Nowadays the temperature, it is nothing but column oven is nothing but it is a chamber of which is controlling the temperature of the column. That's all. Then the detector, HPLC detectors, this detector performance, like character. What are the major characteristics for detector? Like sensitivity, like selectivity, linearity, qualitative information, reliability, and ease of use. So these are the characters which are being taken into account before we choose the detector. So most of the labs, UV visible detector are choice, because most of the components are coming in there. Like nowadays, modified diode array, multiple wavelength, variable wavelength, then the mass spectrometers where we can detect the mass of the compound, refractive index, again it is a kind of universal index, and the fluorescence, light scattering, and the electrochemical, radioactivity, and conductivity. So what kind of the detector is being attached, that is being very. If it is a mass spectrometry, your will be LCMS, liquid chromatography, and the mass spectrometry. So component will be separated, and the mass of the compound will be annotated by the different softwares. How particular. So this HPLC could be, if it's a mass spectrometer, and other you can determine the characteristics of compound using the different detectors. So detector is a component responsible for turning physical and chemical attribute into a measurable signal corresponding to the concentration or identity. In fact, early day HPLC detection was often carried by collecting the fraction and then analyzing. Means whatever just like a liquid chromatography, also column chromatography, we elute different component and then we analyze. So here also collect, in earlier day detector were not there, but until 40s and 50s, the first time online detectors were introduced. However, in 1960, first ultraviolet detector for HPLC was introduced, and subsequent improvement in design led to better sensitivity and improvement of such as variable length and diode array. While truly universal HPLC detectors with GCFID level sensitivity is still elusive, like in a gas chromatography, this FID is very many of different types of detector has been developed that even very successful for wide variety of general use of HPLC and even in specific applications. So this again, just I have already told that it should have a high sensitivity response to it should respond to all solute, having predictable specificity, wide linear dynamic range, and response the increased linearly with the amount of solute, and response unaffected by change in temperature and mobile phase, respond independently to mobile phase, not contribute to extra column band broadening, reliable and convenient to use, the non-destructive of the, it should not destruct the solute, and provide qualitative and quantity information of detected P and the fast response. So depending on detector, nowadays even the detectors are attached with many kind of softwares. So they have their own algorithm, and with that algorithm they detect, like nowadays Kim Spider, MJ Clouds in LCMS machines, or like in a GCMS NIST library were there. So depending on how particularly what kind of detectors are there, so they can be but they should have these particular universal characteristics we should be there. So the HPLC detector selection criteria, what kind of detector we should use, chemical nature of analyte and potential inferences, limit of detection or quantitation required, availability and cost of detector. Sometimes like many we wanted to purchase like true detector, but whatever the fund was given by the DBT, we are because RI detector and the UV detector we want. RI detector we want to detect the carbohydrate like pectin. We have we need to analyze the pectin, but company tell it is our we cannot we cannot able to give within this amount. So ultimately we have to go for the PDA, and we get another column or one or auto sampler out of that. So but see, the cost is also a limiting factor. We wanted to detect, so yes, we can outsource that sample. So cost and availability is also very important. Compatibility of HPLC with HPLC and UPLC system, Many times, many detector cannot be competent. Number of different assays to be performed, and the complementary and orthogonal detection. So these kind of selection criteria should be chosen for what kind of detector we are going to have. So detectors for HPLC are designed to take advantage of some of physical and chemical attributes of solute or mobile phase in a chromatographic process in a one of four ways, like bulk property or differential measurement. Bulk property detectors are most universal kind of detectors. They measure the property common to all analyte by measuring the difference in mobile phase without the sample. One of the most common bulk property detector is a refractive index. And analyte specific detector responds to characteristic that is unique to analyte. Here not a universal kind of thing. The UV detector is most common example of analyte specific detector responding to analyte that absorb the UV light at a particular wavelength. So that's why if the particular component is there, and that will where DNA is absorbing at 250 254 nanometer, or any flavonoid is there which is absorbing at a 270, for example, 270 nanometer. So that will be specific. Sometimes that's why this lambda max is very important. Lambda max becomes very important. The UV detector are usually thought somewhat specific, responding only to compound with chromophores, means color, but the low UV length less than 210 nanometer, where just about every organic compound absorbed. UV detector are actually somewhat universal, and other analyte specific detector include fluorescence, conductivity, and electrochemical. So detectors, so they are like UV visible, photo array detector, refractive, the fluorescent detector, refractive index RID, refractive index detector, conductivity detector, electrochemical detector, evaporative light scattering detector, and the mass spectrometer, as in LCMS, are the high resolution LCMS. So these are there. So if we go through the most visible, like how particularly this is a deuterium lamp, it will generate, and through the grating, the light is being again reflected back into mirror 2, mirror 3, and there is a grating, and then that help into the again reflecting back, and then it is being reaching. So it is a cut filter, the high order of diffracting light filter in a particular source. So this is the basic fundamental. Same thing here, like light source, deuterium or tungsten lump, grating, mirrors, and this like sample one, like energy, like a double beam spectrophotometer. Here we can have a sample cell and the reference cell, and it is detected by the photodiode. So simple Beer-Lambert law, Lambert-Beer law, like how much amount of absorbance is there, it will be proportionate to the molar absorbability, concentration of analyte, and the path length of the flow cell. And this is the energy log of E out, and how much energy is being out and how much energy is being given. So this is the absorbance has a like a normally concentration and absorbance, they have a linear relationship up to this much particular point, and later on it will bend. Ideal situation it should go, but actually this is the situation. So Beer-Lambert law is being routinely used in our in normal UV visual spectrophotometer also. This principle is used, and if UV visible detector is there, the same fundamental principle which is being used in UV visual spectrophotometer, that is being applicable to the UV detector in HPLC. So UV visible detector, how to select the wavelength, which is better, we can see. The UV visible spectrum, the compound has a two absorbance band, like 208 nanometer and 270 nanometer. The chromatogram obtained by UV visible detector at 270 nanometer, the chromatogram obtained at a 200 nanometer. So it is like here in 270 nanometer, you see that three different peaks, they get separated separately. But here it is like it is height is more, but here. So it depends on you, what you want to achieve and how much you want to achieve. Whether first is better or second is better, depending on your situation, you can use either of these. So that is a very important if it has a two. So UV visible detector, what are the advantage? It has a very high sensitivity and relatively robust to temperature and flow rate change and compatible with the gradient illusion. Disadvantage that only compound with UV visible absorption could be detected. Suppose any below UV visible range or like below above visible range like near IR, then we cannot detect such compound. We cannot able to, like many of the aromatic compound which has a in a higher wavelength, they absorb the light. So for that it is very difficult. Additional function, dual wavelength mode and wavelength time program mode and wavelength scan mode is there in a UV visible. We can scan like a particular wavelength. We can scan whole range of wavelength. So that is the beauty about UV. Then the photo array diode, it is a modified form of the UV visible only, like the deuterium lamp sample. Then the grating element, 5 element photo, one element detects one absorption at a particular wavelength. So PDA detector is very common. You can see here absorbance, time and wavelength, like chromatogram and the spectrum. With the spectrum, if we see the same picture in 3D model, it looks like wavelength, time and the intensity. So this is the beauty of the PDA detector. That sorry. So PDA detector could be analyzed samples simultaneously at many different wavelengths. UV visible spectra are useful for the compound detection and checking the peak purity, as well as finding the optimum observation for the compound. UV visible spectra of many compounds could be stored in the spectrum library, which are useful for the compound identification. Whatever the UV visible spectra you have, that can be stored and that can be matched later. Relatively robust to temperature and flow fluctuation, as in UV visible, compatible with the gradient illusion, slightly less sensitive. It has a like simultaneously many different wavelengths, but when it comes to sensitivity, it is little less sensitive than the UV visible PDA. It is more universal in nature compared to UV, but it is less sensitive. So apart from UV, this fluorescence, sometimes we want to determine the compound in a very small amount, which has a, like fluorescence is a type of luminescence in which light energy is released in form of photon in a nano or microsecond, in a very small period of time. So that phenomena is being referred as fluorescence. Light is being absorbed and the energy is being non-radiation transition. It is being released in a photon in a nano and microsecond. So this fluorescence intensity relationship between the fluorescence intensity and concentration, F is equal to 2.3 quantum efficiency into intensity of radiation into molar absorptivity, path length of flow cell, and the concentration. So this is like fluorescence. Depending again, this fluorescence, it is being very specific detector for a specific. It has a very high specificity. So this is the photo. The light is being generated by the not by the normal deuterium or tungsten lamp. It is by photomultiplier tube, and this xenon lamp. Photomultiplier tube is a detector, and it is the xenon lamp is being used, which is being through grating and the flow cell and the EM filters and condensing lens. This is the fluorescence detector. What is the advantage? Sensitivity is higher than the UV visible detector. Selectivity is high because relatively few compounds. It has a very high selectivity because few compound has a fluorescence ability, compatible with the gradient illusion as in a PDA and UV. But disadvantage, difficult to predict fluorescence, greatly affected by environment like solvent, pH, temperature, viscosity, ionic strength, and the dissolved gas. So it is being highly. These are the some of the limiting factor for the fluorescence detector. Then after fluorescence, RI detector, as we see, when the light even passes through one medium to another, it bends. This is the phenomena called refraction. So when tungsten lamp light is being moving, so it will not go in the straight, but it will bend in a sample. Whatever the sample, like here water or any kind of sample is there, it will be bent. That is property called. So using that property, this RI detector, the fundamental principle. So what is the it respond nearly all solute and unaffected by the flow rate. Disadvantage, it is not so sensitive as per other type of detector and could be used with the gradient illusion. So these are the RI detector application, like if we analytical conditions, like column zinc pack, mobile phase acetonitrile and water, flow rate 1 meter, ambient temperature. So if these compounds which are very glycerol, xylose, fructose, glucose, mannose, so these compounds which are very near, their structure or molecular weights are very near. So these kind of components can be easily separated by the RI detector. So RI detector mostly used in the carbohydrate fraction. For our case also we wanted to determine the pectin and its component. So you can see the carbohydrate case, the RI detector has a great application compared to other detectors. Then the other detectors, I will not go much in detail. So RI detector, PDA detector, fluorescence and UV, conductivity detector. The conductivity detector is having this specific conductivity. So I will not go much detail in these detectors. So, then the temperature control detector, conductivity is very affected by temperature. So must keep the cell of temperature control. The column oven can be a choice. Then the conductivity detector, what is advantage? Respond to ionic compounds and suitable for ion chromatography, high sensitivity for lower concentration. Okay, this conductivity detector has a, like if it is a very low in concentration also can be disadvantage. Sensitive to fluctuation of solvent flow and mobile force composition, and non-compatible. It is non-compatible with the gradient illusion. This is a major drawback. Like gradient illusion is very important. For isocratic med, this conductivity detector could be a choice. This application we can see, like this conductivity detector ions, especially now if we have to detect the ions like fluoride, chloride, nitride, bromide in a different ppm, from 1 ppm to like 10,000 ppm. The column could be a SIMPAC, this mobile phase could, so these are the conditions for this temperature and injection. But this ion can be detected easily with the conductivity detector, which is particularly difficult in other kind of detector, no? So these cations also, conductivity detector like sodium anions and cations detection could be possible with the conductivity detector. Then now the electrochemical detector, reference electrode and the others. So principle of electrochemical detector responds to the compound that can be oxidized or reduced, such as phenols, aromatic amines, ketones and aldehydes, which can be reduced or oxidized. So application, phenol compound, general use like hydrogen peroxide, halogen ions and the sugar analysis. These electrochemical detectors can be a choice. Electrochemical detector, it is a selectively relative few compound of electroactive in nature, excellent sensitive for the again lower concentration, just like a conductivity detector. Disadvantage is the sensitive to temperature and flow rate fluctuation. Again, just like conductivity detector, it is not compatible with gradient evolution. Aqueous and other pollen solvent containing dissolved electrolyte are required, and they must be rigorously free from oxygen. So these detector has this limitation. Then the application we can see this, like noradrenaline, adrenaline, or catecholamine, dopamine, which is like you see in the parts per billion, 5 parts per billion in a very small concentration. So that's why I tell, depending on what kind of, suppose these kind of the things, they cannot be detected by other detectors. So noradrenaline, adrenaline, or dopamine, which is being used for the doping test, no? Doping test at a like a sport during sport event. So this kind of component can be detected by this, easily can be detected by electrochemical detector, which cannot be detected by other detector easily. So nowadays this evaporatory ELSD, evaporatory light scattering detector, but this ELSD respond to compound that is less volatile than that of mobile phase. So this ELSD detector is a destructive detector. You cannot recover your sample. That is the thing, but it is a sensitive detector. So this is the like nebulization gas, mobile phase, and the evaporation. It will be evaporated. So once your component is evaporated, you cannot light detection. So what is the application of ELSD? Like in a chemical industry, polymer and surfactant pharmaceutical industry, especially the impurities. What are the impurities are there that can be detected? And in the food industry, like the saccharides, monosaccharide, disaccharide, and the fatty acid could be detected by these detectors. So now, what are the all the components we have come through, like pump, injector, auto injector, and the column. What are the different types of column, and finally what are the detectors? So all these components we have covered. But yes, different components are there, but method development is very important. How we develop, because it is the machine is I just tell in the beginning also, this machine is not a magic box. How we use, how we use, and what purpose you use, and how critically we use, and without basic understanding of chemistry, it will be very difficult. So the common mistakes in the method development: inadequate formulation of methods of goal. We don't know, like many times these phytochemists, no? Botany people think that whatever the components are present in the plant, we can easily separate with HPLC, but it is not so. It will require, even to purify one component. You see the for the recovery of Taxol from the this Texas plant, it takes 14 years to characterize and purify. So it is not easy. Like many times people believe it, it requires rigorous work. So inadequate formulation method goals, little knowledge about chemistry of analyte mixture, use of first reverse column available, whatever the like in a normal university labs and other, and trial and error with different column and mobile phases. So common mistake we do, we start and we do. And these mistakes result in laborious, time-consuming developing project, and method fail to meet the needs of the analytes. So let's see. So HPLC development, before you start, define your knowledge about the sample. What sample is there? What kind of sample? Even suppose in a phytochemistry is there. So like related family, some work is being done, a related genera, a related family. So we can know that what kind of component, whether it is a fatty acid, whether it is a aromatic in nature, whether it is a non-aromatic in nature. So we should know a little bit about sample. Even when first time I send my sample for the HRLCMS analysis at Bombay for oil, then they tell, sir, whether you have some paper in regarding this genre. Then I tell that no, I don't have any publication regarding. Then they tell whether in this particular family, other genre they are being reported. Then I tell yes, and I send that manuscript to that paper. They read it, and they see that how particularly we can design the method, because we cannot use, so similar kind of method they can use for that detection. So then that they have detected very good fatty acids in that oil, we using HRLCMS, and even GCMS we done in our own lab. Now both results are in very coherence. So before we know, we should know about sample, we should define our goals about separation method, and choose the column to be considered. Suppose you are using C4, C18, but actually it requires C8 or C14 up, depending on polarity. So how the result will be there? Then you will think that this HPLC is not working. It is not like that. So we should choose the initial mobile phase chemistry, choose the detector type and starting parameter, evaluate the potential of column for the sample. What is the potential? Whether this can be used or not. Optimize the separation condition, whether it will require isocratic or gradient mode for chosen column, and validate the method to release for the routine level. So it is a HPLC optimization for a particular sample. It is a very time taking. Suppose if it is nothing being known, then it trial and error method, it is your fortune or it is your luck. So like choosing the appropriate HPLC column should be based on both upon the knowledge of sample and the goal of separation. Benefit of this approach include small initial time investment, big time saving in HPLC laboratory. Before, more informal approach to column selection, and more efficient trial and error approach. So what kind of column you have selected, that could be very important. So that's why during our purchase we pressurized the company, and we got 4 different kinds of columns. Yes, we know that in a phytochemistry, but fortunately and unfortunately I leave the Mizoram and I leave that lab. Sometimes I feel so but we got from company 4 kind of looking into this scenario. When this was there, I'm searching lot of knowledge when we are procuring the HPLC, I am reading online lot of knowledge, and then I prepare all this from this. The knowledge of sample influences the choice of column bonded characteristic, like phase characteristics. So knowledge about sampling, stuck if little bit known, not in same genre or other thing. Sample component, number of component present, sample matrix, pKa value of sample component, concentration range, what could be the possible concentration, molecular weight range, solubility, and other pertinent data. So suppose something we are going to do, we should at least something we should know. Then column curve, bonded phase, bonding type, end capping, and the carbon load. So the goals for the separation influence the choice for column particle and the physical characteristic. Goal for separation, like maximum resolution of all component, partial resolution, fast analysis, economy, low solvent usage. See this. Now that's why this method development is very, very important. Nowadays, solvent, it is economically very, very costly. Even if you HPLC grade solvent, you will go, even 5 liter will be cost in many thousand. Apart from economy, nowadays people are talking about green chemistry. How we can minimize the use of solvent, because whatever the solvent is there, again it is going back into nature, causing the pollution. So, until unless it is recycled. So this is very, very important, not as an economic point of view, but the environmental and ecological point of view also. This low solvent uses is becoming a paramount. And the column stability and lifetime preparatory, whether it is a like analytical method or preparatory method, or high sensitivity and other goals. So like column physics, like particle bed, dimension, particle shape and size, surface area, and the pore size. So if we see now again same thing. So depending how whatever the questions we ask about the column, about the solvent, about the thing. So again, like you see, for non-polar, normal phase, for non-ionic polar, maybe the reverse phase may be choice, for ionic, ion chemistry, for water insoluble, maybe reverse phase, normal phase, gel permutation. For water soluble, you may go for the maybe the reverse phase, but ion exchange and the gel permutation. Even you see, for the lower molecular weight, this could HPLC could be very good. But suppose those who have a very high molecular weight, the gel permeation, size exclusion, and gel filtration. So depending on the polarity, depending on the molecular weight, this could be your choice for the HPLC. This could what kind of the things you are going to choose. So procedure method development, optimization of separation in reverse phase, and the calibration method. So we'll give in a small. So this method development for HPLC, what mode, like whether we should go for the reverse phase or normal phase, what column, type of column and temperature, what mobile phase, whether we should go for isocratic and gradient, what buffer and pH, what detector, and what calibration method. So these are the very, very important. So information, yeah, already this we have discussed. Yes. So selecting HPLC mode, method development for most sample often begin with the reverse phase. Ion pair phase and normal phase are the secondary choice. So mainly we go for the reverse phase. Sample exhibiting any type of following characters will require spatial concentration, like high molecular weight sample, such as synthetic polymers or the carbohydrate or period. So obviously this above I just said that for high molecular weight you need to go for size exclusion or the gel permutation. So, mixture of optical isomer. Again, this is very, very like dextro rotatory and levo rotatory, like two enantiomers are there. How to separate this dextro and levo? Because many times see in an antibiotic also levocitrazine. It is not. Levocitrazine is more effective than the its dextro form. So that levocitrazine, the levorotatory form, will be in a pharmaceutical. Suppose you want to, both components are there. So obviously the pharmaceutical company will require levo form more, which is more optically, not only optically activity, but pharmaceutically active. So the mixture of optical isomers, mixture of other isomers, and sample composed of organic salts. So if we see reverse phase in a method development, reverse phase condition that affect the HPLC separation, Like what are the variables? Like column dimension, particle size, stationary phase, which is being given here. I'm not going through the. Then the mobile phase, solvent A and B, how much proportion, buffer, like compound pH and concentration, additive like ion pair reagent and amines, flow rate, temperature, generally 10 to 60 degree, sample size, volume, in generally volume is less than 100 microliter, mass is less than 100 microgram. So this is likely in a reverse phase. You can see the solvents are water and acetonitrile, and it is a variable. Both A and B are variable, and the stationary phase are the ODS like C18 and octa C8. So characteristic for primary HPLC, like the reverse, first choice for neutral and anions, and the ion pair, good choice for ionic and ionizable, and normal phase, good choice when reverse phase or ion phase are ineffective lipophilic samples that do not dissolve well in water and organic mixture. For mixture of isomer, preparative scale HPLC is more important compared to analytical HPLC. So the characteristic of secondary HPLC methods, like ion exchange mode, it is good choice for separating protein and nucleic acid sample related compound. Separation of organic ions, like ion chromatography is being used. Size distribution, again it is a good choice for high molecular weight samples such as protein and synthetic polymers, and GPC for the molecular weight distribution. So these for like in a biological micromolecules are the supramolecules, like these kind of the SPLC are much better compared to them. So improving the separation, resolution value for complete separation of two neighboring peaks should be like it should be more than 1.5. Value more than 2 should be the goal during the method, like this RS value. Resolution, RS means resolution value during development of sample mixture. In case multi-component sample is there, value more than 1 may be separation goal, because it is difficult. So we can see the how to improve the resolution value if multi-component at least more than 1, but our major goal should be resolution value should be 2. So here you see the separation goal, resolution, precise and rugged quantitative, and the separation time less than 5 to 10 meter is desirable for routine procedure quantitation. Resolution 1% to 5% traces. Pressure 150 kg F centimeter square is desirable, and less than 200 kg F is usually essential. Narrow peaks for larger signal noise ratio are desirable. Minimum mobile phase use per run is desirable. So these are the some of the desirable things that in a desirable parameters for resolutions, and being should be considered. And completing the method should be robust in routinal operation, usable by all laboratories. In this regard, HPLC must meet the stringent standard for the precision, accuracy, ruggedness, and the transferability. So method validation is important to complete the method development. It is very, very important. So above parameters is being considered while doing that. So optimization of separation in reverse. What are the optimization procedure in the reverse phase HPLC? Yes, again this RS value. So resolution value for complete separation of two neighboring peaks. Just I tell above, it should be more than. So resolution value will be V2 minus V1 half into W1 minus W2. So you see the peak is not a triangle, but it is simply a Gaussian distribution. It is not a triangle, it is a Gaussian. If it is a 1, it looks like this. If it is RS value is now 1.5, then these peaks are separate. So this is the goal, like RS value is less 0.4, it is more overlapping, but if it is just more above 1 or 1.25, you see the both, there are two components. When RS value is 0.4, they are clumped together. You will think that it is 1, but when at the this RS value increase, these two components, like even isomers, these two isomers can be separated. So resolution, just this is some mathematical thing. Resolution is a function of what? Capacity factor, selectivity, alpha, and the column efficiency. R is equal to 1/4 into alpha minus 1 selectivity, then root of n, that is column efficiency, into the capacity factor k raised to k plus 1. So this is the formula for the resolution, is a function of capacity factor, selectivity factor, and the column efficiency. So the capacity factor is like a retention time of a solute peak, and the column wide time of a solvent peak. So K capacity factor is T1 minus T0 divided by T0. Then the selectivity factor. Selectivity factor is an indication of degree of separation between two peaks. Then the number of total theoretical plate n. So like how particularly equation is this. Then the contribution factor capacity, like you can see the equation with the particularly resolution. Then again the contribution of efficiency, resolution factor, and the factor affecting the resolution. You can see inject, initial, and inject. Then if we see the alpha and n, then the two peaks are getting resolved. The K, alpha, and n, these are the factors which are very important for the how to increase the n. So H is equal to length of column divided by n. So volume, each column has is a optimum flow rate and the linear. You can see the linear velocity. So when we talk about the optimum flow rate, to be good column efficiency, these are the some of the preferable setting. 0.6 ml per minute, 0.8 ml, 0.1 ml per minute. These are the preferable settings. Then the again the smaller particle size, use of smaller particle size is being desired, and again these flow rates are being there. Then how to increase the K? By decreasing the percent of organic solvent. K2 is 10 is a preferable. So the inefficient separation, if K is less than 10, the long running time, broad band cause, low sensitivity, and poor percent. So it should be like as much as lower. So if we can see 2 to 10 range, it is much better. The solvent optimization, like sample component also, if you can see here 100% methanol is there, you can see it affects the K value, and you see only one peak. But you see it contains like 2,6 dinitrotoluene, 2 nitrotoluene, 4 nitrotoluene. They are very near this component. But if suppose methanol and water 80 raised to 20 is there, and the value of K will be less than 0.6 to 1.7, then you can see the different component and be separate. So again the solvent optimization become very paramount. Solvent optimization is very paramount in distinguishing or in differentiating the peaks of the very near or similar kind of component. So the relationship between K and the percent solvent, the log of K is equal to A into B percent into B. A and B are the coefficient. For small molecules, a 10% of increase in the percent of organ solvent, B decreased by the K of the every band by factor about 3. So here you can see peak 1 and peak 2. So these factors, relationship between K, the logarithmic equation which tell the relationship between K and the percentage organic solvent. And then how to improve the alpha? By changing the mobile phase and composition, pH, concentration of buffer, column temperature, and packaging material to C8, Cn. So these, depending on what kind of alpha our change improve, we can change the goal of alpha. If we want to improve the alpha, then we need to consider for these components in the HPLC procedure. The change in the mobile phase, you can see the critical pair C and D, critical pair. So depending on, although one organic solvent cannot provide good resolution, combination of mobile phase has a possibility to provide good resolution. It is a critical pair for has been seen. So what exactly solvent? What are the one solvent cannot be a kind of universal thing. So in a routine kind of thing, you can see methanol and water, tetrahydrofuran and water, methanol and tetrahydrofuran and water. So these components are there. You see in the first pair, this is the resolution. In second, this is the resolution. But if all threes are being mixed in a particular amount, you can see A, B, C, D are Definitely, well, here in this critical pair, A and B are very well resolved. In this critical pair, C and D are very well resolved. But all these three components are being there. But again, for that you need like a HPLC with the binary or quaternary. So if you want to do again the column and the pump you require, so such kind of the thing is very important. Like effect of mobile phase pH on separation of ionic compound, like you see the benzoic acid, sorbic acid and methyl parabolic analytical condition 1 ml per minute ODS column and 10 millimolar of phosphate buffer, 70% of acetonitrile and 20% of nitrogenitrile. So you see the pH 2.6, pH 4.6 and pH 7. So depending on the like maybe the 4.6 may be very important, higher and lower compared to this. So pH has a impact on the especially separation of ionic compound because pH is being generally affected by the ionic stage. pH of mobile phase, for example, the containing acid or basic component retention time may vary with pH, and the adjusting pH of mobile phase is very important to get good repeatability, and the buffer solution must be selected correctly. So this relationship between it is a normal Anderson-Hasselbalch equation. You can see the K is the of organic acid and amines vary with the pH. And we can see the pH pKa 4.6 point. So in the range of pKa plus or minus 1.5, a linear relationship between K and the pH is exist. So here associated and dissociation. The precaution in pH adjustment, when pH of mobile phase is very close to pKa, the target of the target sample pH adjustment will be very close. If pH and pKa value are very close, we should be since pH of a mobile phase will influence the K, and the normally pH which does not affect K should be selected. So pH should be, while selecting the pH, we should be very cautious. And pH and pK of solution is a normal, like pH is has a direct impact, direct relationship with pK with log of a ion and the cation. So this is the pH pKa of organic acid for buffer, for phosphate buffer, for acetate buffer, concentration and pK value. So these when we are doing for ionic separation, these kind of the pA and pK value are very important. How to prepare buffer solution? pH calculation is accurate for low concentration of buffer. For high concentration of buffers, the pH of must be measured with pH meter in first preparation, and second preparation of buffer pH may be not necessarily measured with pH meter. pH can be always used for pH meter should be always used to confirm that, because that pH value will have an enormous impact on separation. So again we can see ODS and phenyl command. Effect of packing material on characteristic, like see the benzoic acid, sorbic acid and methyl paragma. If analytical conditions are same, but if we use phenyl column, this is the separation, and if we use ODS, this is the separation. So depending on the packing material, the separation is going to vary. Then addition of organic solvent, addition of co-solvent beside methanol, acetonitrile, tetrahydrofuran, sometimes effective to get better separation. So like if we use the co-solvents, then like ethyl ether, isopropyl ether, dioxane, methoxyethane and cyclohexane oxide, around 10% can be added to normal mobile phase for the reverse phase. So you can see acetonitrile and water, 10% of ethyl, addition of 10% of ethyl acetate has a better resolution compared to acetonitrile and water. So again, this is the obvious that sometime addition, it is not always, but the sometime addition depending on situation, addition of the organic solvent may have a positive impact on the peak resolution. Use of gradient elution method. If two neighboring peaks cannot be separated by any condition using isocratic elution, these peaks cannot be also be separated by condition using gradient elution method. It is necessary to confirm isocratic mode can provide some condition to separate two peaks when gradient mode is selected for even faster analysis. Gradient elution may be helpful to reduce the analysis time with sacrificing. So for such kind of the thing, gradient elution is there. So optimizing the gradient method. If still further optimization is required, change of long gradient time change of faster flow rate, use of longer column. Sometimes we use the column length also become very, very important, and the change of organic solvent, methyl acetonylene and hydrofuron, and the problem of gradient, poor inadequate column equilibrium, time required for the column regeneration is usually about equal to gradient, and the long calibration is required in case of only water or buffer as in initial mobile. So it is advisable to begin with the gradient with 5% organic or greater. Use of auto sampler to minimize the variation in equilibrium time is now being highly suggested. We can see here problem 2 in gradient, low grade of waters, because many times this we use for HPLC milliQ HPLC grade water, or sometimes buy HPLC grade water. 1 liter of price also costs 600 rupees. 1 liter water of the HPLC grade. So this water has some impurity which can as a ghost peak. Use of water change a longer wavelength which may not show the absorption impurity. It is advisable to begin with the gradient of 5% organic or greater. Calibration method in HPLC. There are different calibration methods like external calibration method, internal calibration method, standard additive method, and correction normal. Calibration is very, very important actually. Even we do experiment, we prepare standard in the normal procedure also, suppose enzyme assay or antioxidant assay. So whatever the, like suppose standard we are using, we prepare different concentration, and we prepare standard. So every time we prepare standard. So the calibration external like same way we do for normal procedure with the spectrophotometer, here also external and internal calibration is a paramount of in a HPLC method. Same way. So like you see the external calibration, preparation of standard stock solution, like different, like tenders to maybe 1. So we can keep on diluting working standard, and this will give a increasing concentration. So if we get the standard calibration curve, peak area, like with the increase in the concentration, there will be increase in the absorbance. We can calibrate through the peak area. We can get the what is the exact concentration. So simple line equation, y is equal to ax plus b. A is a slope and y is intercept. So on intercept, we can easily value of b we can easily. So here maybe 200 pp this in a peak also we can do. Then the intercalibrate same way, like stock weighing standard and the internal standard. So we can mix the standard, internal standard, increasing solute and the constant. So you can give like internal standard analysis for value. Internal standard you can just see. Internal standard calibration for value. So it's the target area and the target concentration and internal concentration. Internal calibration, again the same concept. So I'm not going much in detail, like this same way. So this is the way. Like advantage of the recovery of the pre-treatment process can be estimated, and the disadvantage of internal separation is slightly difficult. So disadvantage of internal calibration, it is difficult to look for internal what compound we are going to use. That is very, very important. Chemical structure of internal standard compound is similar with one that of target compound. IS sample not existent in actual sample. So that is very for using that has a further limitation. So external standard calibration separation is not difficult. Injection error will directly influence the qualitative result. Injection error can be eliminated. Recovery in the pre-treatment procedure can be estimated. Separation is slightly difficult. Difficult to look for standard internal standard calibration that is one of the very, very limiting factor in the while choosing the internal standard. So these are the standard additive in the calibration, original sample, and we add the standard original additive. So suppose 1000 ppm respond to this, so obviously the 70 ppm will respond. The standard additive method in a calibration. So this is this accurate quantitation, sensitivity, expected range, and the linearity. This is the we expect in HPLC that like it should be detection response should be sensitive concentration of solute, sensitivity expected range and the linearity. This, whatever the method we talk about, this is the accurate for accurate computation. Sensitivity, expected range, and the linearity is a very important. So if we talk scope of HPLC again, in my very beginning, I talked that these are like in pharmaceutical, in biochemical, in the food product, in the industrial chemicals, in the forensic chemistry, or in environmental field and chemical field. These HPLCs have a vast range of application in day-to-day. Not detection at a particular concentration is very important. Like in a blood also, even if somebody has consumed something, any poison for society also, how much it has consumed. So doctor can make the according to the they can provide the drug, or how much, suppose like during the even in a road also, how much alcohol somebody has consumed. So we can that easily detect, or any kind of how much toxin, or how much additive is being there. Like in a food material also, there are permissible limit, but if it is beyond the permissible limit, that can be easily detected. So these are the some of the compounds, like in a pharmaceutical, like antibiotic, like sedative, like steroid, like analgesic. It can be easily detected. Like in cosmetic, these are the compound. Like in the antihypertensive or antioxidant, gallic acid, which is being phytochemistry lab. These kind of compound very important. And then biochemicals like amino acid, protein, peptide, carbohydrates, biochemicals like lipid, enzyme, hormones, fatty acid, biological amines, and the food products like mycotoxin, the flavor and fragrance, and the dietary supplements. Nowadays, dietary supplements become very important. So even natural products are being sold with the everything is herbal, but we don't know what that herbal is being containing. It may be sometimes it may be life threatening. People are selling because many of the drugs which are being sold without any medical their examination. What could be their they are being sold without any having medical test or animal test. Their safety, their toxicity. So that dietary supplements need to be checked very rigorously. What kind of compound? Sometimes maybe good compound, bad compound. Even the vitamins. Many of the vitamins they are being, even vitamin A, D discovery, they are being done on the HPLC. For that they are being awarded Nobel Prize. The food products like coloring agents, sugar, antibacterials, preservative, artificial sweetener, residual drugs, and this like forensic chemistry like hemoglobin, azodide, cannabis, diamorphine, and this like environmental field. Nowadays, the role of HPLC becoming very paramount, detecting the organic ions, organic acid, a lot of pesticides. This pesticide residue is highly toxic. In a food chain, this how much pesticide is seen, antibiotic and pesticide, they are being now detected in the bloods, in the milk, mother milk. So this toxic residual toxicity in long run is detection, because sometimes what the water we are drinking from what area, whether they are what they are following appropriate regulations or not. Even in river also, what how much effluent they are being discharged. So suppose if that effluent can be detected, they are beyond the limit. So this pesticide in the phenolics compound or any kind, that are the things. Our lab also, in our lab also we are just detecting this apigenin, marangine, risperidine using standards. So that time just we initiated, but I got the job in my near hometown, so I leave my dreams there, and I came here in University of Allahabad to join. Now again there is a new beginning at University of Allahabad few months ago. But in phytochemical things, we DBT project we are working, and during working for 3 4 months only, I came to know about the HPLC. Whatever the knowledge in that during period I acquire about HPLC, how the, because for optimization of HPLC is very difficult. So what are the things that are required? So I read through internet, I consult some expert, and I came to know that yes, whether we are going even with my research scholar, we are now many for other two flavonoids, the peaks are very near. So we are working on column temperature, varying the column temperature, and even for pH we are going to devise, but unfortunately we leave that project, that will also resign. So whatever the knowledge, little knowledge I acquired during my this research project, I share with you. So I am very thankful. So thank you very much.
So I really like to thank Dr. Sanjay for such an extensive and such a very, very informative and excellent speech. So I should like to invite all the participants in the groups. If you have any questions, then you are obviously welcome to ask him. I will invite all the participants again that you be ready with your questions. If you have any questions, please ask him. Or I will prefer that Dr. Sanjay, that if you can share your email ID in the message group, then any person can be contacting with you.
Yes, yes, sir. Yes, sir. I will just write here in message box.
Okay,
So
so you
I
share
share
your
my
email ID.
yeah, I will share my email ID. If anybody has any query later on also, they can always welcome.
So here, so in the message, actually, you can see that Dr. Sanjay has given his email ID. If you have any questions regarding HPLC, or if you have any questions regarding his specializations, you are always welcome to contact with him. And I'm still asking to all of the participants that if you have any queries, please ask him. Otherwise, we are going to wind up the session. And I shall be really thankful to Dr. Sanjay to have his precious time to share in the seminar. And as you are a very dear colleague, a very dear friend, and an outstanding scientist, we shall be very much happy to have you in the near future also.
Thank you so much, sir. Thank you, participants, for your patience hearing. So you are always welcome for any suggestion, any help. Because in scientific community, we need to help each other. No? We little know about many things, and we like so with sir also I gain many knowledge, no, while discussion. So keep on keeping in touch. Interacting in science is very, very important. You don't know, like you see the chromatographic technique. Also, the Mikhail Tesvet, when he developed this technique, people are not knowing. In calcium carbonate, he separated the chlorophyll and carotenoids, and during his lifetime, people have not recognized because they are using high concentration of calcium carbonate, and they are not able to reproduce. Later on, 10 years later, Australian scientists, they discovered the methods for chlorophyll and carotenoids, and they separated, and they recognized that scientist. Now we are Mikhail Tesvet is a very pioneer worker in the field of the chromatography. So knowing from each other is a very, very important parameter, because of very little we know. And whatever, I'm not a chemist, I'm a botanist, pure botanist working on a pure plant biology area. But I learn because there are three colleagues working on the project. One is from chemistry, one is from medicinal plant. I'm from forestry, but I'm very much interested. So that's why I learn and I share today. Thank you, sir. Thank you so much.
Botany is always related to chemistry.
Yeah.
Because if you're not having a perfect knowledge of the botany, then you shall be not able to understand chemistry also.
Yes.
Because as you know, Dr. Sanjay, that whatever is my field of specializations, that when we are inventing a new polymer, then we have to be aware about that, about the high molecular weight polysaccharides,
Yes.
right?
Yes.
So the high molecular weight polysaccharides and to interact the polymers to give a novel polymer, that is very much related to botany and chemistry.
Yes. Yes.
So you are having an extensive knowledge in botany. You are an outstanding scientist, and whatever the views you have shared in your seminar, I think everybody are enriched with your knowledge.
Thank you.
And
Thank you so much.
wish you all the best for your career. And I wish you, and I will tell all the participants that you be in touch with Sir in his email ID to solve any problems in botany and chemistry. And we are soon going to live stream your presentation in our Facebook site, in our college Facebook site, and the participant will be having the further opportunity to have a look on their to have a look on the slides in their leisure timing, and I'm sure that they will be reverting back to you in your email ID.
Okay. Okay, sir.
So, Dr. Sanjay, I'd like to thank you once more time, once again, and it's a wonderful to have you on the last day of our seminar. You made our activity really successful. Thanks a lot to you.
Thank you so much, sir. Thank you so much for giving me an opportunity to interact with the people from diverse background. Thank you so much, sir.
Thank you, sir. Thank you, sir. So all of the participants are requested to leave the seminar. Okay, sir. Goodbye then.
Bye, sir.
Goodbye, sir.